Uh, Welcome everyone to the Data Science Seminar Series. It is a pleasure today to have Professor Caroline Uhler to join us virtually today. Professor Uhler is currently the Henry and Grace Doherty Associate Professor at the Department of Electrical Engineering and Computer Science at MIT, and is also a core faculty member at the Institute for Data Systems and Society. She's also the co-director of the Eric and Wendy Schmidt Center at the Broad Institute between Harvard and MIT, and it's a member of the Laboratory for Information and Decision Systems, also at MIT. Professor Uhler holds a master's degree in mathematics and a bachelor's degree in biology from the University of Zurich, also a master's in education in mathematics from the same university, and a PhD in statistics from University of California at Berkeley. Before joining MIT, she spent some time as a fellow of the Simons Institute and holding postdoctoral positions at the MIA and ETH in Zurich. She was also an assistant professor at the IST in Austria. Professor Uhler has won many awards, including the NSF Career Awards and the Sofia Kovalevskaya Award from the Humboldt Institute, the Simons Investigator Award, and the Sloan Research Fellowship. So Professor Uhler has many research in different areas that span signal processing, biology, control systems, machine learning, causal inference, and it's an honor for us to have her here. So I'm sure her work is going to be of a lot of interest for all our audience. So without further ado, thank you for being here, and you can share your screen.
Thank you so much. I just started sharing, and let me just go to full screen. Okay, does this look okay?
Yeah, sounds good.
Great. Okay, so thank you so much for this introduction, and in particular for the invitation to speak here. So I wanted to talk about some recent work, as was just mentioned. So we've been working on causality quite a bit. And so during COVID-19, the group really wanted to work on something together and see how the kinds of things that we have been developing could be used in this crisis. And in particular, we've been working on the problem of drug repurposing. And so I want to tell you a little bit about that, and in particular, how this relates to causality and how we've been using it. Okay, so let me just first take a step back and talk about what is actually the problem and what does drug development mean in this in the particular context of COVID-19. So if we're thinking about, when we have these kinds of situations like what we've seen, it's quite an urgent situation, and so we all know that developing drugs from scratch can take very long, and in fact most drugs fail. So that means that in this particular situation, probably the most viable approach is just drug repurposing. So that means you're looking at drugs that are already FDA approved, and then you try to find drugs that might also be effective in this new context. So that means that you don't have to go through these safety trials anymore. So these have already been passed. You have to go through the efficacy trials, of course, because you need to show that they are also effective in a new context. So how do drugs generally work? So drugs are usually inhibitors. So they bind to a protein so that it cannot perform its downstream role. And so if we're thinking about it, and in particular here, I'm going to, want to suggest that taking a causal approach is in fact important. And so I think that already comes out here with this definition of what most drugs do. So they're inhibitors, so they bind to a protein, so that means it cannot perform its downstream role. So that means you need to know what the downstream targets are, right? So it will only have an effect on, so you need to know something about the underlying regulatory network, because this whole drug will only have an effect on whatever the genes are or the proteins are that are downstream of the particular protein where this drug is actually binding to. And maybe already let me, if I get to like what are actually computational approaches that have been used for drug repurposing or also drug development, most drugs have been, of course, developed experimentally, completely experimentally, but there have been drugs that have been brought all the way through that have been developed, in fact, computationally. And so I just want to talk here about two main approaches, and I'll be using both in the talk. And so this one main approach, and here you'll see how causality is actually missing and how it has been used, is using undirected graphs. And I know here many people are studying graphical models and network models. And so there are these very large-scale network models that are available in biology for how proteins interact with each other. Okay, so that's a very large-scale network where your nodes are proteins and then the edges correspond to protein-protein interactions. And so a standard approach is in fact to take like additional data from the disease context. So in this case, COVID-19, and for example, look at gene expression. And so that means that you have additional data on the nodes, right? And then look at, well, which nodes are differentially regulated in the disease or differentially expressed in the disease. And so as compared to the normal state, right? And so maybe you can color them in red if they're down as compared to the normal, and maybe in blue if they're up as compared to the normal. And then you have all these other ones like gray and whitish that don't show any difference in terms of disease versus normal. And so now you have this huge network on thousands of different nodes. And so now you look at these colors and you try to find like areas in the network where there is a lot of misregulated stuff, right? And so that's those would be the subnetworks that you may want to concentrate on. These are also known as these disease interactomes. Okay, so these like subnetworks that are really misregulated by the disease, there definitely something is happening. And so standard approaches would then take these subnetworks and then try to look for nodes that are targetable by a drug and that are very central to these different kinds of subnetworks. Now, you can already see that there is, of course, a problem with this, right? That, if your network actually really looks like here in this middle picture, where, you're targeting a node that is very central, but all of these edges are actually pointing in, well, then this drug cannot have any impact on all of the genes around it. And so this drug is going to be pretty useless. However, if the network looks like this, right, where all of these edges are actually pointing out, then at least the drug has the potential of being useful. Of course, these interactions have to be going in the right way, etc., and have to be pushing you up if you're down, etc. But at least, here you have a potential of actually having a drug that is useful in this particular disease context, because it does have a possibility of changing the nodes around it in the right way. Okay, so this already shows why causality is probably important when you want to actually look at and identify drugs in different disease contexts. So that's one way where causality enters. Now, I also want to talk about a second approach, computational approach that has been used quite a bit for drug screening. And so that's the following. So it's known as signature matching. Okay, and so I'll also explain it a little bit more on the next slide. But very intuitively, so you have data of the normal state, as we were using before for this network approach, of the normal state and the disease state. So you can define something that was, or in this direction, something that means what the disease signature looks like. Okay, so that's just so here, say we're measuring data often at the beginning, and when you're studying a disease, you often get data which is maybe, say, like gene expression data at the cell level. So that's like a 20,000 dimensional vector. And so you can define the disease signature as this vector which points from the normal state to the disease state. That's your disease signature. And now what you would like to find is drugs, right, that point you back. So now what you can do if you have large-scale drug screens is try to figure out what is the signature of each drug. And then you would prioritize drugs that are most anti-correlated with a disease signature, because they are potentially possible to actually move you back to the normal state. So that's what is known as signature matching. So matching the disease signature anti-correlated with the drug signature. Okay, so if we think about that, so also I want to connect that to causality, but let me just actually show these kinds of data sets and how they look like, and what you could use for doing this signature matching approach, which is actually one of the data sets that is often used for this. So what you see here is a very large scale, actually publicly available drug screen, where many drugs have been, and other kinds of perturbations have been applied to different kinds of cancer cell lines. So around 70 cancer cell lines is what you see here, and really thousands of different perturbations, among them 800 FDA approved drugs, but many other perturbations, also non-approved drugs, etc., are here. And so this was a screen for cancer. And obviously, what you don't want to do is have to redo this whole screen, right? Whatever time there is like a new disease context, anyways, you'll not be able to like always test like all these different kinds of perturbations in every new disease context that you're looking at. And so what you would really like to be able to do is somehow use this data and be able to, from here, predict what the drug signature will be in your new disease context, and then do the signature matching. Okay, now what is important to note is that a drug can have very different effects in different cell types. So it is not a trivial thing of like, I just look at whatever one cell type and then I'm like, yeah, okay, so this is the effect of a drug. And so it will just have the same effect on this other cell type. Okay, and also what is important to note is that, and this is just, this representation of all of this data that is available here, is that, and here it's colored by different cell lines, in this case, or cell types. So you see that the cell types, which are shown in different colors, so the different cell types have a much bigger spread than the different perturbations. So here what you have in black is all of the different drugs applied to this particular cell type. And believe me that, these are actually the drugs applied to this particular cell type. And here you see that the drugs apply to this particular cell type. So the variability of what you can do with a drug is small as compared to differences between cell types. Okay, so in fact, if you just want to predict the effect of a drug, it's actually quite difficult to beat just a very simple estimator, which is just predict the effect, the mean of this particular cell type. Okay, just so that we know that we have a particular baseline. So drugs cannot really move you outside of the space of your own cell type, right? Which kind of makes sense. So there are only very small perturbations. So actually estimating the effect of a drug is quite a hard task. Okay, so this data set, as you see here, there is a whole lot of white space. So this is a partially filled matrix, quite a sparse one. And it's really a tensor, because here you have all of the perturbations, here you have all of the cell types. And one of these black entries means that in there, we have a 1,000 dimensional measurement. Okay, so in humans have 20,000 genes, as I mentioned. So, often you would measure all 20,000 genes. Here, what they decided, just also, it is very costly to do that, is to just choose like 1,000 very well selected genes. And those are the 1,000 genes that they're always giving the data for every one of these entries. Okay, so this is known as L1000 data. So that's what this is. Okay, so it's a tensor. So whenever you have one entry in this direction, you'll have the full row, right? Because then you get to measure all of these genes in one measurement. Okay, so from this data, what we'd like to do is, use all this data where we know what these drugs are doing on some cell types, and try to predict what the effect will be of these drugs on this new cell type, namely SARS-CoV-2 infected lung epithelial cells. So that's what we want to actually study. Now, why is this a causal question? So in fact, this is known as causal transportability. Okay, so something that has been studied quite a lot by Elias Badenboim, Julia Pearl, etc. And I'll also have this on the next slide. So it's, and if I frame this in a different problem, then it's clear that it has a lot to do with causality. So often this causal transportability has been studied in the literature of policies. So if you have the effect, if you know the effect of a policy in one city, right now, what you would like to do is also predict the effect of this policy in a new city that you have not yet seen. That's causal transportability. So I know the effect of an intervention in one context. I would like to, from there, predict the effect of this intervention in a different context. Now, drugs and cell types is exactly that, right? A drug is an intervention on the system. It only has an effect downstream. So now you know the effect of a drug in some cell types. These are the contexts. And what you would like to do from this data is predict the effect of this drug in a different context, namely a different cell type. So really what this problem of like filling in of this matrix completion problem is actually not your standard matrix completion problem like a Netflix problem, right? It is a very different problem because it is a causal problem, because our rows and our columns are different, right? Our rows are, if you are familiar with causality, is like the do operator, whereas our columns or conditioning is like the standard thing in Netflix is like your movies, but here it's not people, but it's actually drugs, which are interventions. So this is a causal matrix completion problem, and also known as causal transportability. Okay, so this is the problem that we'd like to solve. So I think it is clear that for both approaches, whether you're looking at it from this network approach or whether you're looking at it from this matrix completion kind of approach, you should take, there is causality to be taken into account. Okay, so let me talk first about this one here, and then I'll get to the networks approach all the way at the end. But I'll mainly be talking about this matrix completion problem. Okay. So let's talk a little bit about if you would just wear a machine learning hat, what would probably be your first approach of how to actually solve this? And down here, I have the references to Barenboim and Pearl's work on causal transportability. Okay, so if I was just wearing a machine learning hat, then, probably one of the first approaches for actually trying to solve something like this is something like a style transfer. Where I'm sure you've seen all these pictures of nice, Yosemite in the winter and Yosemite in the summer, right? Just the style transferred over without actually retaking the picture. So, and that's what I'm showing here is, if you have, and I'm showing it using autoencoders. Often, people use GANs for doing this, but I'll be using autoencoders in the whole talk just because we do have a theoretical framework for autoencoders. Okay, so what is an autoencoder? I'm sure we're all familiar with it. It's just two neural networks, right? When one neural network maps you into a latent space, and then the other neural network maps you from the latent space back to the original space. And these neural networks are trained just based on reconstruction error. Okay, so whatever you put in, whatever comes out should look very much like what you put in. Okay, so that's your reconstruction error is what you're actually trying to optimize. Now that will lead you to an informative latent space, right? Because it has to keep track of everything that is important in order to reconstruct whatever your input images was. Okay, so now if you have like a whole lot of faces, like for example, I have here, and maybe some people like to smile and others don't like to smile, you can train this autoencoder using all these images that you have. And now what is style transfer doing? And this is actually quite interesting that this often works quite well for these imaging problems is, you have like this person. So here is a person who I've never seen smiling. Okay, so there is no smiling picture, but I would like to generate a corresponding picture when this person is smiling. So actually what works quite well is you take some other people where you do have a neutral face and a smiley face. And that, of course, I can look at in the latent space where they land, right? And so this vector here corresponds to adding a smile. At least for this particular person, it corresponds to adding a smile. Now, what is quite interesting is that you can actually take this vector, move it up here to this person here where you want to add a smile, right? Now here comes, this is a dot in the latent space, but an autoencoder allows you to generate a corresponding image. And so I can turn this dot here in the latent space into an image. And what you see as out comes actually this particular person smiling. Okay, so that's style transfer. So I can, just transfer a style over to a new image, and out comes that particular image with this particular style. Now you can think of this as maybe also, and this in fact, has been tried, and I know also drug industries have in fact tried this already. Like for example, AstraZeneca has tried this, where you could say like, well, maybe I can do something like this for drugs. So I know, what the effect is of some drugs on some cell types, right? So now then the question is, well, can I then just do the same thing? I take in the latent space the vector corresponding to adding that drug, and I just move that drug effect over to a new cell type. Now in this paper, which is actually a really nice paper by Fabian Theis' lab, they have shown that something like this can actually work for some cell types. Okay, so they've done it, I think there were like just a couple of perturbations and a couple of cell types, and it seemed like it worked quite well actually. And so that was quite nice. Now, of course, because we have this huge cell type, this huge database, right, you can actually test generally whether something like this can work or not. And this was also seen by AstraZeneca. This generally, unfortunately, doesn't really work. Okay, and so how do we test whether this works? Well, we just train our autoencoder on all this data, right? And then here you can take like, let's take just the first couple of cell types which have the most drugs applied to them. You see here that, most of these entries, they're actually the least sparse ones, right? So I can look at what the effect of a drug will be, right, after in the autoencoder for that particular cell type. And I can look at the same drug, like whenever I have two entries here, I can look at the same drug on the other cell type. And this approach will work if the effect of the same drug in the latent space is aligned, right? So otherwise this effect will not work because all I'm doing is taking this vector and moving it over. Right. So how I can, measure whether such a method can work at all is just by looking at the alignment of these vectors or the correlation between these vectors in the latent space versus in the original space. And it works if in the latent space they're more aligned than in the original space. And so that's exactly what I'm going to show you, which I'm doing now on this data set. Okay. So I'll just do exactly that on this data set. And now, okay, so these are the pictures, and I want to go through the data a little bit carefully. But first, so what I'm going to do is show you what happens when you do this in your standard autoencoder. So this is the autoencoders that was used in this paper that I just showed you. And as your standard autoencoders, so autoencoders have been developed for dimension reduction, right? They're similar to PCA. It's a nonlinear version of PCA, where you start with something high dimensional and you want to find a nice low dimensional representation of your data. Now, what we're going to propose, and this will sound very crazy right now, but I'll go through the theory of why this is actually something interesting, is we're actually going to use autoencoders to embed into a higher dimensional space than what you're starting off with. Okay, so I'm showing you, and then I'll give some intuition for why this might be interesting. So I'm starting off with our 900 dimensional vector, right, this L1000 data. I'm going to embed it into a higher dimensional space here, and then of course back to our original space. Now you'll tell me that this doesn't make, and this is why nobody has done these, never used these overparameterized autoencoders. It doesn't make sense, right? Because there are enough parameters here to just learn the identity map. I mean, right? I have no bottleneck here, so there is nothing that keeps me from just learning the identity map. Now, what I want to already show you here is that these autoencoders actually don't learn the identity map. And then I'll give you the theory of what are the functions that are learned and why did we even have the intuition to actually use these kinds of things for this particular problem. So it really came from the theory. But let me first actually just already here, I can show you that they're not learning the identity map because that's quite easy to show on these examples. So here I'm exactly looking at two cell types that have the most drugs. The results are the same, and I have for all cell types, I have it over here, but let's just look at two cell types. Okay, and so I'm looking at the correlation. So I'm looking at all of the drugs that are shared between these two cell types, so that have been applied in both cell types. I'm just looking at the correlation of these drugs. Okay, and so what you see here is and this is the correlation in the original space as compared to the latent space. Okay, so here you see it's a lower dimensional representation. We're going from 911 to 100 dimensions. Okay, and you can play around with whatever latent space dimension you want, and you'll basically always get the same results. And what you see here is that basically you just get the diagonal, so you're not doing better than in the original space, but you do have a little bit of an enrichment up here, which is good, right? So this means that you have a little bit of alignment. Now, what we see down here is, in this case, this is in fact the overparameterized autoencoder. And in fact, we can prove that the more overparameterized you are, the more you're actually going to go to a step function. Okay, so that everything will in fact turn out to be more aligned. Okay, and what you already see on this picture is that certainly it did not learn the identity map. Okay, that's definitely clear. It did not learn the identity map. So, whenever you're doing something like this autoencoder, it is actually learning something interesting, and definitely something interesting for this particular application. Now, you may say like, well, let me just do something very simple. Like, let me just do PCA and say, take just the first two principal components as my representation. Now, what is interesting is that you actually get very similar pictures, right? So you might tell me then like, well, I mean, then why don't I just use the first two principal components, since I'm going to get my drugs be aligned in this latent space? Well, the problem is that when you're just using your first two principal components, or any PCA with just the lowest principal components, you're going to get alignment by throwing out basically all of the information in your data. So that's a different way of getting alignment, is just by throwing out all of the important information in your data, right? And that you see here by not being able to reconstruct basically anything in your data set. So that's not how you want to get alignment, right? You want to keep as much information as possible in your data and still have the effect of drugs be aligned so that you can actually translate between them, because otherwise you also lost all of the cell type information, everything basically. And so you see that, of course, when you're overparameterized, you don't lose any information. I have perfect reconstruction. I have training error 0, right? And of course here, this is fine. This is like okay reconstruction, right? Which is just what you're getting with your standard autoencoders. Okay, so it seems like these autoencoders are actually learning functions that are interesting for this particular application. It is an odd autoencoder, right? It is training error will be 0, but it is not learning the identity map, although it could. And so what I want to do now is actually go into the theory of what is the function that is learned by these overparameterized autoencoders. And then I'll get back to this at the end, okay, of like how we actually use this for drug repurposing. So it's quite clear. Maybe let me just make the story finish it so that we don't forget it, right? So what this all allows us to do is, in the latent space, we now, from here, we get the signature of a drug, right? What this gives us is, in the latent space, we can now get the signatures of drugs that are transferable across different cell types. And so that means that now I can look at the disease signature in the particular cell type that I care about, right? And look at which one of the drugs is most anti-correlated with this disease signature. And based on this anti-correlation is how I'm prioritizing and going to rank drugs. And this is what I'll go to show you at the end of what actually happens and what comes out. Okay, but let me now go into the theory of like, what are these autoencoders? Why do they actually learn functions that might be interesting? And why don't they learn the identity map? Okay, and so for this, let's just actually look at the simple setting. So first, let me set up the problem. So I have my N training examples, and they live in, say, RK, and I'm doing overparameterization. So overparameterization for me, all it means is that training error can go down to 0. Okay, so you have enough parameters to get your training error down to 0. So one way of doing that is, if your N training samples is less than K, right, the space where they're living in. Okay, so let me just write it out like this. So this, just for simplicity of actually just writing down one particular example, I'm going to look at this setting. So this is your standard autoencoder loss, right? Where, this is my autoencoder, my neural network. This is what comes out at the end of the neural network. This is what I put in. So what I'm trying to minimize is the reconstruction error. Okay. So, okay, so this is what I'm trying, this is the optimization problem that I'm trying to solve. Let's just, to get some intuition, let's look at the simplest setting where I just have one sample, okay? So one sample and say linear. So my neural network is just a linear map. So then what this looks like, and hopefully I can write here, yes, is just A times X minus X, right? In 2 norm, and I can do a square or whatever. And I'm looking at the argmin, and A is in RK times K. And this one here is just a vector, right? This is in RK times 1. Okay. So this is the problem that I'm looking at, right? A very simple problem, A times X minus X. So obviously I can get this down to 0. But the thing is that there are many solutions, right? There are many solutions, matrices A, that can make this be equal to 0. The identity matrix being one of them. But there are many others, right? There is a rank 1 projection onto this vector X. There is a rank 2, there is a rank 3, etc. There are many, many solutions to this problem. And so really the question is, what is the solution that the autoencoder is learning? Right, so if you use, and so for all of our theoretical results, we're using gradient descent, initialized close to 0. So of course, when you're just applying it, you're going to use some stochastic gradient descent or whatever, whatever optimization algorithms you want to do. But you usually initialize these, actually close all your weights close to 0. So this is preserved there as well. Okay, so our theoretical results will be looking at gradient descent. Okay, so this is a well-known result that if you're looking at your linear setting and you just use gradient descent initialized at 0, what actually you're getting as a solution here is your rank 1 solution. Okay. So now the question is, of course, what happens when you have non-linearities here, right? And in general, and then what is the solution that is learned when you actually have these non-linearities here and when you don't have just one training example? Okay, so let's get a little bit of intuition, and let's just do that by actually training, your standard UNet autoencoders, which are some of the well-used and prominent autoencoders that are currently being used. And just to get some intuition, I'm just going to train it on one training example. Okay, so these cute little bunnies is the training example that I actually trained this UNet autoencoder on. And so to get some intuition for what is the function that is being learned, well, I can just plug in, right? The autoencoder just takes in some images, right? In this case, images, because it was trained on images, and outputs images. So what I'll do to try to interrogate what the function is that the autoencoder learned is I'll just put in some random images and see what comes out. Well, and so what you see here is that no matter what you put in, even if you put in just Gaussian noise or some nice little figures like this, no matter what you do, always your cute little bunnies come out. Okay, so this is actually more extreme than just learning a rank 1 function, right? Because here we really learned the point map. So whatever you put in, it was actually matched to the bunnies. If it was rank 1, it could have also been the negative of the bunnies, or any kind of linear combination of these bunnies, right? Which could also have been just a flip of the sign. And in fact, that's what you see in the linear setting. So if you train this using a linear network, then in fact what you get out is the whole line. But this is more extreme, okay? So when you have nonlinearity, this is a very extreme thing that happens. Now, I do want to, sometimes it seems like, well, people would say like, this is maybe what I had expected. But let me show you that maybe this is not what one should expect, or it is still, I mean, still should be kind of surprising that this is what happened, because generally this is not what happens. And in particular, there are infinitely many solutions, right? So the question is, why did they choose this one? Okay, so let me now show you what actually happens when you do a shallow network. So UNet is actually quite deep as when you just have one training example. So when you do a shallow network or when you do a linear network. So I basically already told you actually what happens in the linear setting. And here I'm actually showing it to you. I'm using two training examples so that we see very cleanly that it always learns a linear combination of the training examples, okay? So whatever you put in, it will actually always map to a linear combination of your training examples. And so that's what you see here. And you can prove that. That's actually very easy to prove. Okay, so let's look at a shallow network, so not as deep as your UNet autoencoder, just to see that actually something interesting also happens with depth. Okay, so not just nonlinearity, but actually depth is somehow also important for the function that we got on the previous slide. So if you don't do deep, then what you see here is that basically whatever you put in, right, the same thing comes out. So it seems to learn something like the identity map. Now, just a word of caution. So there are papers that suggest that these autoencoders actually can learn the identity map. Now, this is wrong. So although it seems here that what we're actually learning is the identity map, it is not. And I'll show you on the next slide that this is not the identity map. Okay, so but it is interesting, right, that if you go shallow, then in fact, what it seems to be learning is something very, very different than what we saw here, right? Here it was the point map basically, and here what we see is actually something like the identity map. Okay, so it depends on depth. And so let's analyze how it depends on depth. And in fact, so it's a super easy and simple idea. And I'm going to show you to you on the next slide on how we are actually interrogating these, what the function is that is being learned and how we can very easily show that this is not the identity map. So what you do, right, is an autoencoder maps from whatever it is where you start off, RK to RK. Well, so then what I can do is just whatever comes out in the autoencoder, I can plug it back in, right? And let me just iterate the map and see what happens. And then you'll see that this is not yet. If it was the identity map, then this would be stable, right? I would just keep getting out this little horse here. So if you do that, in fact, and so here we trained, these autoencoders on like 500 images. And so what we did is, instead of just starting in a random point, we just started in training examples where you add a whole lot of noise on top of it and just see where you're iterating to. You can also take random examples and, we'll have proofs afterwards, but like you can plug in whatever you want. But then what you see is like, yes, if I would have just gone one step here, you would have basically gotten out something that looks the same as before, like kind of what you see here, right? So whatever you're doing, you get in one step, you get to the same. But then if you keep iterating it and keep iterating it, et cetera, what you see, what ends up is actually you're ending up in a training example. Okay, so this is not the identity map that was learned. And in fact, the functions that are learned are highly contractive at the training examples. Okay. And so this is, if we visualize this in 2D, this is how these basins of attraction look like. So these were all of our training examples here, these little stars in 2D. And then, we just made a grid to look at, if we start somewhere here and we just iterate the autoencoder, where are we going to converge to? And so this is quite interesting, right? These basins of attraction look actually quite interesting. And, this will be an open problem. I think for later, it's really important to understand, like, what are these metrics that are actually learned by autoencoders, right? And, how do these basins of attraction actually look like? So I think there is still a whole lot of research to be done here because it's really important for applications in terms of, different nonlinearities would probably lead to a very different kinds of metrics in the latent space. And so for different kinds of applications, this is really what we actually have to understand. Okay, so that's the intuition. Okay, so a very simple idea of like, well, let's just analyze what happens when you actually iterate the map and try to understand in this way what the function is that is being learned and then prove something about that. Okay. So what the hypothesis is that it is highly contractive at the training examples themselves, right? That this is the map that is being learned, and that this contractivity, I mean, this is now also clear, right? If I go back here, this contractivity should depend a lot on depth, because if I do really high depth, then, you just need one iteration and you're already at the training example. Whereas, if you do very shallow, very little depth, right, then you have to iterate your autoencoder many times until you end up with the bunnies. Okay, so that's how depth will enter in, and we can actually see things like this. First of all, it is very easy to actually memorize training. I mean, so to build networks that can memorize many training examples, and I'll talk a little bit about what I mean by memorization on the next slide. But for now, memorization just means that when I iterate them from any starting point, there is they are attractors. Okay. And so, you can build like quite small networks. They can already memorize 500 examples. And we now have a proof that, large enough networks will memorize any number of training examples. Okay, so that was this, and you can actually build them, and we can build them very easily. Okay, so this was the proof and the theorem that was in this PNAS paper. We now have it like generally. So this is for one training example. We now have it generally for any number of training examples. But so this is what this theorem says, and so it's like actually very constructive. So what is it what we're looking at? So we're just proving it here for one training example, and what we're saying is, okay, so you have some autoencoder of some depth and some width. Okay, then you're using some nonlinearity and some initialization, and here there are conditions on it, but all of these conditions, your standard nonlinearities, will all satisfy these conditions. So these are not crazy conditions on the nonlinearity. And so now what this says is that, well, what we did in the proof is actually we just computed the eigenvalue, the maximum eigenvalue of the Jacobian at that particular training example. Okay, and so this is now a formula which we have, in which depends on the depth d and the width k and the nonlinearity. And what we're proving here is that you can choose your depth and width, so you can choose it large enough so that this eigenvalue will always become small. Eigenvalue and absolute value will always become smaller than 1, meaning that it is in fact an attractive fixed point. So that's what we proved in here, that in fact, whatever we have been seeing on the previous slides is actually happening. So we don't have it in this proof for any number of training examples, but there it was for 1. Now we do have it for any number of training examples. So basically this phenomenon is what we proved, right? And in fact, even with less depth, depending on the nonlinearity, right, you actually do get something that is contractive at the training example. So you might have to iterate and then you get back to your training example. So certainly what it says is that definitely this is not learning the identity map. Okay, so let me do a couple of remarks here, because I think that is always important here in this case. So there are always questions about generalization, and there are usually questions about memorization. Okay, so first of all, let me start by, maybe talking a little about generalization. So first of all, we know that, deep convolutional neural networks, right, they can interpolate training data even with random labels. Okay, so this was known for a long time, etc. Then there is always a question about generalization. But let me first just make a difference about what I mean by interpolating and what I mean by memorizing, because those are like often used as the same, but I would actually like to make a difference between them. So, here is a function that goes from R1 to R1. So let's look at autoencoders that are super simple. They go from R1 to R1. Okay, and my black dots are my training examples. I'm overparameterized, so definitely at the training examples, I have loss 0, meaning that I'm learning a function that goes through the training examples. That's what is called interpolating, right? So if I get my training loss 0, that's what it means to be an interpolating solution. Now, to me, this is not a memorizing solution, okay? Because, here so this is like some random function, right? That is interpolating. This is not what the autoencoder learns, but I would not call a function like this memorizing because I cannot recover the training examples from this function, right? There is no way if I give you this function that you can tell me that this dot and this dot and this dot were the training examples. So to me, this is not memorizing a training example because you cannot retrieve the training example. However, these are the functions that what we're showing are actually learned by autoencoders, right? So what we saw when we have very deep autoencoders is that actually these step functions are learned, right? Where it's just a point map onto the training example around, some basin of attraction, right? So whatever you put in, one of your training examples comes out. So this is what so it's certainly interpolating, but I would also call this memorizing, right? Because, well, I can actually retrieve my training examples by just putting in random noise, for example. Okay, so this one here is definitely memorizing, and that's the difference that we're making. Now, this one here is exactly what we're seeing, what these autoencoders, these overparameterized autoencoders are learning when you are not super deep, right? So they learn functions that are contractive at the training examples, right? But they can still be, and in fact, and this will be interesting, they can still be close to the diagonal, as we've seen, right? In this example where we had like a very not deep, but a very shallow network. So they can be, and actually you can show it, they can be arbitrarily close to the diagonal, but in fact still contractive at the training examples. Okay. And so that's, and this is also again memorizing, because I can just iterate the map and outcome my training examples. So, you know, I can retrieve the training examples from these maps. So that would be the difference between interpolating and memorizing that I would like to make, and that I think, you know, we should be making if we want to use these terms in the right way. And then the other question is always about generalizability. And, you know, yes, our training error is 0, but we know from the standard classification neural networks how you can, you know, be overparameterized and still actually generalize well. And there is a lot of literature there. Now, this one here is on the autoencoder setting, and the first thing that I want to bring up is that I think generalization has not really been defined for autoencoders. So what does it mean to generalize well for an autoencoder? So people would sometimes use it as like learning the identity map. Well, I mean, if that to me does not seem like the right definition, at least because, you know, if you want to learn the identity map, I don't need to train anything, right? I mean, the identity map, don't train, like just, you know, put out whatever your image is that goes in. So that shouldn't be the right definition of generalizing, I think, at least. But I think we should have there should be a discussion of what should be the right definition of generalizability or generalizing if we're autoencoders. And, you know, one definition could be, and, you know, who knows if this would be the right one, but and this is in fact how we're now using our autoencoders when we're using them on different applications. We're actually going by this because it seems to generalize well and now I can talk about generalizing in the particular problem instances that we're working on, in the biological problem instances. So what we're looking at is to be close to the identity map, but still contractive at the training examples. Okay, so that cannot be the identity map because it wouldn't be contractive at the training examples. So we would say that a definition might be of generalization good generalization for an autoencoder is that you're close to the identity map, so as close as possible, but still contractive at the training examples. You should be contractive at the training examples, right? Because if you have something that is very close to a training example, you definitely would want it to remain close to the training example or become even closer, and certainly not like in this map over here, where it can just move you like super far away, even if you're super close to a training example. So that seems to be a property that you may want your autoencoder to have. And in fact, Joshua Benjo, and just, you know, so people have actually looked at adding regularizers for exactly this, for getting maps that are contractive at the training examples. So they certainly thought for many different applications early on that, you know, you may actually want to have autoencoders that learn these maps that are contractive at the training examples. And so what we show here is that you don't need any regularizers, right? Just overparameterize, and these are the maps that are actually learned by autoencoders. So don't make your problem like super difficult, right? By adding all these regularizers, just add more parameters in the latent space and you're done, and you're actually getting these self-regularizing. And the more overparameterized you are, the more self-regularizing you become, right? Because you're becoming more and more like the step function here. Okay, so that's the whole intuition for the maps that are being learned. There is a whole lot of things that we still need to show and prove, and here is just some intuition. And, you know, these plots are maybe, I mean, we've looked at so many plots, this will not convince you. I think you'll have to look at your own plots of this as well. But it seems like what happens, and I'll just summarize this, what happens with increasing depth is that the mean of the eigenvalue shifts more towards 0, or the mode shifts more towards 0. And what happens with width is that the variance of the eigenvalues, training examples, becomes smaller and smaller. Okay, so from this, what we have been now always using in our autoencoders is, you know, if we define something like generalizing that you want to stay close to the diagonal, but still contractive at the training examples, well, then what do you want? Well, you want your variance over the eigenvalues to be very small, right? So let your width be very, very large. But then you certainly don't want to go too close to 0, because, you know, then like, you know, whatever example you put in, you always get out the training example. You certainly don't want to be there, right? You want it to be like very close to the diagonal. So that means you want your eigenvalues to be just a little bit smaller than 1, right? But very close to 1. And so we usually just add enough depth, right? So that we are contractive at the training examples, but still very, very close to the diagonal. So this gives some intuition, but there are a lot of, here there are no proofs, right? So there should be a lot of proofs in terms of that this is in fact what is happening, but this is at least, empirically what we've seen, and now also what we're using when we're actually training our own autoencoders on the particular problems that we're interested in. Okay. So I'm just going back to this particular problem that I started off with. So this is where I left you, right? When we started off going into actually looking at this overparameterized autoencoder a bit more in detail, and actually saw, what it is in fact learning. And so these are the autoencoders that we're using for this particular application, right? Which seems to actually align the causal effects in the latent space. Now, just to give a little bit of intuition, so, there are, for us, different ways to think about it, why this is actually happening. And, it's not so clear that any of our intuition was actually the right one. So, so we still don't have the real proofs of, how to actually connect these overparameterized autoencoders to causality, but at least a little bit of intuition I can maybe give. And maybe for this, I should actually go back. I should have had that little image. Maybe I go here. So, if you want to, so what we need, right, is like a latent space where the causal effects are aligned. Now, if you go into a smaller dimensional latent space, then you're crumbling up your space even more, right? So that's certainly not a good thing when you want to align vectors. So that's the first intuition, is that maybe, you should actually have a large dimensional latent space if you want to have things still remain aligned, or make things aligned with each other that were not aligned before. So the bigger the latent space, right, the easier this will become. Certainly not when you're going to a small dimensional space where everything will become even more crumbled up than what it was. That's intuition one. The second intuition really came from the theory, where, and this is again something that should be proven, but what we proved, right, is that that zero-dimensional points, that you are becoming contractive at zero-dimensional points. Well, we were thinking that maybe you're also going to become become contractive, not just at zero-dimensional things, but maybe at lower dimensional things. So maybe even one-dimensional things and two-dimensional things, etc., will remain aligned. And so if you want to make directions more similar to each other, maybe it will actually also be contractive at these different kinds of directions, so lower dimensional manifolds. So there's no proof for that, and we don't know if that's actually right or not. But that was at least just to tell you of why we even looked at these kinds of autoencoders, and then when to prove that what the functions are that they're actually learning, and how useful they are in fact for all applications. We're now always using our overparameterized autoencoders for anything we're doing. Okay, just to show you how this whole story ends. So now, this is the disease signature from the latent space. We can get all of the drug signatures. All we're going to do is align them. And then, we get a ranking of our drugs out. And what is actually quite nice is that these drugs, the ranking, is actually all of the drugs that you're getting out are just coming out of, 3 different families, basically, or 2 different families, actually, of drugs. And just a little bit of context that this is actually, that this maybe makes a little bit more sense, is that, if you don't, and what we did here is the disease signature is not just disease, but it's actually disease and aging. This whole paper is about, how in aging, the disease signature might be more pronounced, and we see that the outcomes are actually very different. So we're taking the aging disease signature. If you don't take aging into account, in fact, here you get all kinds of drugs out. So that's quite interesting, that aging actually seems to be an important filter. And I'll get back to that again when we're actually looking at the drugs that come out. Now, okay, so you get here a whole list of drugs out. Now, many of these drugs are cancer drugs, so they have a whole lot of targets. And of course, with it, they'll have a whole lot of side effects. So what we wanted to look at is a little bit more causal in, if we have to prioritize among these drugs, or even, figure out among all of these targets that we're finding here, right, that these drugs target, well, which one might be the causal one? Okay, so which one among all these targets that these drugs target, these genes that these drugs target, which one might actually be the causal one? So now going back to this, how I had explained it right at the beginning, what we would like to now find is, so we have all these drugs, they target some different proteins, right? And we would like to figure out which one of these targets is actually the most upstream of all of the differentially expressed genes. So that's all we did. So we've worked for years now on actually how to learn causal networks, right? How to learn causal networks from a mix of observational and interventional data. So that's what we've applied here. And what is actually quite interesting, and so we did robustness tests in terms of looking at very well, very related kinds of different cell types, and seeing that you actually both in the lung, and seeing that you actually get the same network out. And this is quite interesting, that no matter which one of these you take, you actually always get the same target that is the most upstream, which is this RIPK1. Okay, and again, if you don't take aging into account, RIPK1 actually falls out. So it is basically, not upstream anymore. So again, something quite interesting. And this paper actually just came out when we were writing ours, which is quite nice, is that actually this RIPK1 is one of the proteins that can directly bind to SARS-CoV-2. Proteins. So another thing that is really nice about this protein, which of course we didn't add into this analysis at all, but at least is a little bit of first, there is no experimental validation, and experimental validation has to come, but at least, some suggestions that this might actually be an interesting protein. Now, here we also have a hypothesis of what this protein actually does. And I knew nothing about RIPK1 before. I've never heard of it, but it's actually a super interesting protein. It has two domains. Depending on which one you're actually activating, it leads to very different downstream pathways. Okay, so in one pathway, it actually turns on immune response, survival pathways, etc. Basically, what you see in individuals where it goes okay, right? And in many young individuals. And then in the other, if you turn on the other one, it actually leads to all of the death pathways. So apoptosis, necroptosis, and then, the downstream effects are fibrosis and blood clotting, etc. Exactly some of the things that we actually see in the disease. And so what could be happening is just, and we had papers on this, that like maybe the mechanical state of the lung matters. And so depending on in which state you are, actually different kinds of proteins. So the same virus infection, you have like the same protein, but actually different domains that are getting activated in this protein. And so it might actually, just by having the different domains activated, might lead then to very different, we know leads to very different pathways. And this might maybe explain, the differences in the outcomes. Okay, so we do have a mechanism, a suggested mechanism. Now, of course, there is a whole lot of validation to be done. And what is really exciting is actually that a new drug that is specifically targeting RIPK1 actually is just entered phase 2 trials for COVID-19. So it is really interesting to then see what actually happens with this drug. So with that, let me end and just say, so we think often about transport problems in biology. I talked about the problem of transporting perturbations from one cell type to another cell type. But there are all kinds of other transport problems of transporting from one perturbation to a different perturbation. So to a new drug, like from old drugs, predict the effect of new drugs, say, or to transport between different kinds of data modalities. So from sequencing, transport, say, to imaging, or between time points, right? You have, in cancer, you're looking at snapshots in time. You may want to predict the state of these cells at a later time point when you cannot yet measure it. So a whole lot of transport problems in biology that we have been very excited about. Many of them actually causal, because you have these interventions. Okay, with that, let me end. So I think it is important for drug discovery to actually have a principal causal framework. This led us to, all this theoretical work that we've now have been really excited about in terms of autoencoders. And these autoencoders might actually shed a lot of light on what is happening on neural networks more generally, even in the classification setting, although we care in biology much more on the representation learning setting of things. There is a lot more, open questions. I already mentioned some of them down here of like, what are these metrics that are being learned? What are these basins of attraction? How do they depend on the nonlinearity, et cetera, et cetera. Okay, and with that, let me end. I talked a lot about actually, in fact, work of all of my students. So they all got together, and in fact, the current students, they all got together and really worked on this together on the SARS-CoV-2 project. We're really excited about that. And on the theoretical aspect of these autoencoders was mainly Adit's work together with Misha Belkin. And then on the biological, everything that I showed on the biological side is in collaboration with Shiva's lab at ETH Zurich, a longtime collaborator on the biology side. Okay, and then with that, thank you all. And of course, thanks also to funding. Without it, wouldn't be possible. And I'll just go back.
Okay, so great, thank you. Thank you for this amazing presentation. It's always very exciting to see all these new connections between signals and biology, right? Like, and how can we leverage all the tools that we have for other problems to tackle these important issues with biology. So now we can go ahead and for questions. So if you have questions, please you can either type them on the Q&A or the chat, or just raise your hand, and I'll give you access to the mic so you can ask the question yourself. So please go ahead and yeah, you can raise your hand. While we wait for other people, maybe I'll ask a small question. So in the slide, when you have the theorem and the memorization plot, I wonder if there is some relationship here about the smoothness of the function that you're learning, right? Because precisely this idea that you mentioned, that yeah, and the leftmost one is pretty much like a noisy version. We cannot recover the training samples from that. But as you go to the right, the function seems to be more and more smooth, right? So is there some relationship with approximating smooth functions in this case, or imposing smoothness on the function you're approximating?
Yeah, so I think this is an interesting question, and we just know that these are not function. We don't know anything about smoothness of like what is the function's smoothness that you're actually learning, etc. So, and I think it is an interesting question, and we're sometimes asked this kind of question, and yeah, I mean, there is already, like, if we're looking, where was it here? If we're looking at these regions, I mean, there would be even simple things to prove, like that they're always connected. These basins of attraction, etc. But yeah, like saying anything about the function class more than what we'd said, that it is contractive at the training examples, would be very interesting, right? So yeah, so all we know is that we're learning something that is contractive at the training examples. Yeah, but definitely a lot of important open problems that are still there. Yeah.
And from the experiments that you have run, scale wise, like how large do you need to have your data sets, like not like training and testing, just to have like good results? Do you have any intuition about the size of the data samples that you should have for this problem?
Well, okay, so these results, I mean, there has also been another paper, right, which says exactly this. Like, also these results, I mean, it's already clear from here, and there was a paper, I think maybe last year or something, that, you should throw away data. This is what this says, right? At some point you should throw away data, because if your computational resources are limited, you should be throwing away data, because you definitely want to be overparameterized. And so if you cannot be overparameterized, then you should use less data. So that's what this maybe shows, which is maybe a little bit odd, right? But in some sense it makes sense, because you definitely want to be far away from this limit of, like, the problem is always like this data size equal to number of variables. But in fact, what this all shows is that if you're on the other side of like more parameters, than much more parameters than samples, then you're actually doing quite well. But you don't need big data. I mean, you don't need big networks, right? You don't have to be that much overparameterized even to memorize like a lot of training examples. So this is a very small, I mean, depth 10 is like, with 1000 is a very small network.
Right. Yeah. Okay. Yes, there is one question from the audience. Maybe I can just give you, so you can ask the question.
Yeah, I do see it.
Yeah. Hossein, you can ask the question with the mic if you prefer. Yeah.
Okay, can you hear me now?
Yeah.
Yep.
All right, perfect. So my question is, what kind of guarantees do we need? For example, let's just talk about some super extreme cases where interventions are like, one is make sure that the patient eats breakfast, and the other one is a super invasive treatment. So then the question is, okay, we have observations of results of one intervention for all the patients and one intervention for some of them, but then how can we actually guarantee whether we can solve this causal transferring problem or not?
Yeah, so okay, so I mean, this is all a great question, right? So first of all, and I think I didn't point this out, but for causal transportability, there are necessary and sufficient conditions which were given by Pearl and Barenboim. And So the problem with those conditions is that they require you to know the underlying causal network. So if you know the underlying causal network, you know that the intervention affects this node, and you know that the differences between the two settings, between the two contexts is this node, then the network describes to you whether you have causal transportability or not. Okay, so there are necessary and sufficient conditions. Now, in biology, we don't know the underlying network, right? So we cannot apply these things, but there are, right? So I could, we can try to translate what they. So we did this, in fact, in one paper where we're like, so we seem to be getting causal transportability for this particular problem. So why is it? Well, maybe we can go back to their papers and try to hypothesize what the networks are and how the interventions interact with what the cell type definition is, and therefore say that, well, maybe this is why we get causal transportability, right? And so there is this result, and the question is, how do you actually merge the two? But we can just test it, right? We just tested in these particular in this particular data set, but it has to do with how cell type is defined as compared to where the drugs act, for sure, right? Because there are necessary and sufficient conditions, so it will not always work. Yeah.
Okay, so just to make sure, we need to have some common knowledge, some common background knowledge incorporated in designing such experiments, right?
No, you don't need any common knowledge. It's just, so we can test when it works and doesn't work, right? That's what you have your training test for. But there are, if you have the underlying knowledge, then there are necessary and sufficient conditions, which is done in this work by Barenboim and co-authors.
Okay, yeah, I get it. Thank you.
Great. So Chia Yi has a question, so go ahead and ask.
Hello, I want to ask a question that is not, as related to, what the biology example. I want to understand this framework in terms of solving other problems. Like, for example, if we consider a cryptography problem that we have like encoded messages, and then if we apply this kind of autoencoder to the encrypted message, can we learn something latent about them? And then that would be, something about the security, learning.
Yeah. Okay, so I think I have it here as one of the conclusions. So there is a problem with this, right? So this also suggests and tells you that you should not be sharing, for example, trained autoencoders between hospitals, because, right, I can always recover all of the training examples. I can just start off with a random one, and then I just iterate it, right? And then out comes a training example. So this has severe privacy issues. But I would, well, yeah, so for privacy, this is not a good thing. For the applications I care about, it's important that it's contractive at the training examples, because otherwise you're learning your random functions. So anyways, we should have expected, right, that autoencoders are probably not great for privacy issues. But yes, so yes, there is consequences for it, and we can actually recover the training data.
So yes, I want to go toward like even more in a cryptography or confidentiality view. That is, like applying this kind of method, can we actually decrypt something that is being encrypted?
Yeah, but anyone can, so maybe not great.
I mean, like more efficiently.
Oh, I see what you're saying. So the memorization kind of issue. So this is actually something, so here, right, it's like basically this, like how much do you actually need in order to somehow memorize like different kinds of examples, or whatever your codes are, etc. So this is an interesting question, and there has been this work, in fact old work in neural networks, on trying to figure out what are neural networks that are used for memorizing examples or images or codes or whatever you want. And this is a very different framework for memorizing. So it's a completely new way of memorizing that people haven't considered when it was there. And I think there is a lot to be done here. We did write it out, for associative memory, etc., but we didn't do more than that. So I think there is a lot to be done in terms of, and I wrote it at the end, in terms of actually understanding, this is a biologically plausible mechanism for associative memory, and how efficient is it, etc. I think all of these are great questions.
Thank you.
Thank you.
Great. So let me ask one more time. If there are any questions, please raise your hand or type them on the chat.
Cool.
Yes, there is one. Go ahead, Guha.
Hey, can you hear me?
Yep.
Yeah.
Well, actually, it was on this slide, so it's good that you ended here. Your last figure there was kind of intriguing to me. I was curious if you had any insights about these kind of architectural decisions. Like, I guess you show on the bottom, activations seem to have some kind of effect. Is there anything you found so far about how they affect, let's say, the autoencoders and their ability to, or how they change their representations?
Yeah. Yeah, so we don't know. That's why this is an open problem. But we definitely know that the nonlinearity has an effect in how many and how quickly you can, like how much depth and width you actually need in order to memorize training examples. And it already comes up in our theorem, of course. But, that's why based on the theorem and based on, that's why you see here, we use these trigonometric activation functions. They're very good at very quickly memorizing, getting you contraction at the training examples. And you would also find it in the theorem as well. But yeah, so they have an effect. I think there is a lot. We only have looked at it in terms of like, how quickly it leads to memorization. But
Yeah.
I think it's a good question. Yeah.
Thanks.
Thank you.
Great. So we are a little bit over time now. So let's finish now. And again, thank you, Professor Uhler, for joining us today. We hope to have you in person at some point in the future.
Thank you all very much. This was fun.
Yeah, thank you. And thank you all for attending. And have a good rest of the day and of the week. Okay.
Thank you.
So, thank you. Bye.
Bye.